Hello everyone. Today I will talk about elements of pharmaceutical development. This is covered under the annex of the ICH Q8 pharmaceutical development. In today's session I will talk on pharmaceutical development, quality target product profile QTPP, critical quality attributes CQAs, critical material attributes CMAs, risk assessment during development, design space, control strategy, and product lifecycle management and continual improvement. Definitions as per ICH guidelines related to elements of pharmaceutical development. QTPP, Quality Target Product Profile. It is a perspective summary of quality characteristic of drug product that ideally will be achieved to ensure that desired quality, taking into account safety and efficacy of the drug product. Critical Process Parameter CPP. It is a process parameter whose variability has an impact on a critical quality attribute and therefore should be monitored or controlled to ensure that the process produces the desired quality. Critical quality attributes CQAs. A physical, chemical, biological or microbiological property or characteristic that should be within an appropriate limit, range or distribution to ensure the desired product quality. Critical material attributes. A physical, chemical, biological or microbiological property or characteristic of an input material that should be within an appropriate limit, range or distribution to ensure the desired quality or output material. Critical material attributes definition is non-ICH definition. Control strategy. A planned set of controls derived from current product and process understanding that ensures process performance and product quality. The controls can include parameters and attributes related to drug substance and drug product materials and components, facility and equipment operating conditions, in-process controls, finished product specification, and the associated methods and frequency of monitoring and control. Design space. The multidimensional combination and interaction of input variables and process parameters that have been demonstrated to provide assurance of quality. Working within the design space is not considered as a change. Movement out of the design space is considered to be a change and would normally initiate a regulatory post-approval change process. Design space is proposed by the applicant and is subject to regulatory assessment and approval. Pharmaceutical development. In principle, pharmaceutical development is based on that the product should be designed to meet patients' need and intended product performance. This strategy may vary from company to company and product to product. There are empirical and systematic approach or combination of both for development, but today let's understand about systematic approach of pharmaceutical development. The minimum elements of systematic approach of pharmaceutical development are predefined objective, product and process understanding, process control, sound science, quality risk management. There may be more elements than presented here, and that depends on the development and product characteristic. The systematic approach provides a greater understanding of product and its manufacturing process. This scientific knowledge can be applicable for the flexible regulatory approach. The regulatory evaluation is based on the scientific and the risk data. This data should be submitted with each application. Predefined objective. This consists of defined QTPP and identified CQAs. QTPP, this includes the intended use in clinical setting, route of administration, dosage form and its different strength, drug delivery system, container closure system, bioavailability, pharmacokinetic characteristic, and identified CQA for the drug product qualities such as sterility, purity, stability, drug release like immediate release or extended release, product and process understanding. Product characteristics have impact on final product quality. Identifying CMAs such as particle size, particle distribution, bulk density, and CPPs like speed, temperature, humidity. These are basically controlling the process to achieve the desired product quality. And established functional relation to links CMA CPP to CQAs. Process control. Develop process control strategy and justification of this strategy to control the process with the predefined criteria to achieve product quality. Sound science. Prior knowledge, previous scientific data or information, design of experiment which gives a statistical and mathematical tool to perform the experiment in a systematic way and analyze the data efficiently. Science-based pharmaceutical development to design the quality into the drug product. And finally, quality risk management. This is the risk-based approach, very much detailed in the ICH Quality Guidelines Q9. This flow diagram shows the link between CMS, CPPs, and CQAs. The selection of input materials containing right material attributes used in the pharmaceutical development. These input materials are controlled by applying process parameters during development process to achieve the identified CQAs to meet the predefined objective and QTPP of product. These QTPPs can be assay, identification, content uniformity, degradation of product, dissolution, microbial limit, sterility, plant uniformity, particle size for inhalation product. Let's consider the example of tablet. The selection of the input materials with the right attributes such as particle size or granule size. These are the CMS for the tablets development. By using these right materials and controlling the process by critical process parameters such as mixing speed, time, humidity, or temperature to achieve the intermediate CQS like blend uniformity and the final CQS like content uniformity and assay. For clean sterile liquid development, the solubility of input material and the selection of right solvent to achieve the stable solution. These are the critical material attributes. The CPPs such as maintain sterile boundary by selection of right filters, positive pressure of vessels, completion of successful SIP cycles, flow rate, mixing speed, temperature, and maintain the closed manufacturing system integrity. By selecting the right input materials and CMS and applying the CPPs during the development process, the sterility can be achieved of the sterile liquid. This is the final CQA of the product. These are the common CMS, CPPs, CQAs of each size reduction step of pharmaceutical unit operation. Each input material carries their own physicochemical characteristics. Each input material has different particle size and shape, moisture content, flow property, adhesive or binding property, and so on. To achieve the CQAs such as uniform particle size, moisture content, bulk or tap density, these input materials require further processing. The selection of processing operation is based on the input material attributes. Various critical parameters apply during the operation to achieve the defined CQAs. In this example, the selection of granule or ribbon milling controlled by applying CPPs such as mill type, speed, blade configuration, blade types, blade orientation, screen size and screen type, and feeding rate. Error in selection of input materials with right attributes and less or no control on the process parameter may resulting in inconsistent CQS results. Here is another example of relationship between critical material attributes of input material and the critical process parameter related to quality attributes. Controlling CMAs and CPP are the most critical parameters during the pharmaceutical development. In this example of blending and mixing operation, blend uniformity is the CQA. Particle size and particle size distribution are potential high-risk input material attributes. Mixer load level, number of revolution are potential high-risk critical process parameter, and operation room temperature and relative humidity is external element which may impact on the CQA. Right attributes material may reduce the variability in unit operation, and operation can be well controlled by applying critical process parameters. Let's understand the impact of departure from the input material attributes and process parameter. Variation in particle size and size distribution can potentially affect a product manufacturability, efficacy, quality, bioavailability, and shelf life. Particularly in API, it may potentially impact on blend uniformity. This can be the high risk for the other CQA like content uniformity and assay. High or low mixer load level may cause the improper exposure to blending machine parts to achieve the proper mixing. Number of revolution speed may generate heat and create sticky cake for heat sensitive input materials. Less mixing time may not enough to uniform mixing, and longer time may create potential segregation of input materials. External elements such as operation room temperature and humidity can be potential high risk for the hygroscopic input materials. Quality Target Product Profile (QTPP) is the basic for the pharmaceutical product development. QTPP provides an understanding of what will ensure the product safety, quality, and efficacy of a specific product for the patients. QTPP describes the design criteria for the product and should therefore form the basis for development of the CQA, CPPs, and control strategy. Important points to be considered while setting up QTPP during the development stage. It should be intended to clinical use. It's about the product label claim that the product is to be used for. Route of administration, drug delivery system, dosage form. For an example, oral, intravenous, topicals. Important for drug stability, bioavailability, and effectiveness. Dosage strength. To achieve desirable therapeutic effect, control overdose and its related side or toxic effect. Container closure system. For product stability, child-resistant packaging. Therapeutic drug release or delivery system. Attributes affecting pharmacokinetics characteristic. For an example, dissolution, aerodynamic performance. Drug product quality criteria. For an example, sterility, purity, stability, and drug release, like immediate or extended release. Here is the example of QTPP elements for immediate release tablet. As discussed earlier, QTPP element covers the dosage form, dosage design, route of administration, strength, pharmacokinetics, stability, drug product quality attributes, container closure system, labeling, or alternate method of administration. In this example, justification is also provided for the each QTPP elements. Critical quality attributes (CQAs). CQAs are affecting product purity, strength, drug release, and stabilities. CQAs can be product specific, such as aerodynamic properties for inhaled product, sterility for parenterals, adhesion property for transdermal patches. It can be material-specific attributes such as particle size distribution, bulk density. Prior knowledge and defined QTPP are important to set CQAs. Potential CQAs can be modified after deciding the formulation and manufacturing process of final product without compromising product quality, safety, and efficacy. CQAs can be evaluated as product knowledge and increasing of process understanding. Quality risk management tool is useful to prioritize the list of potential CQAs and impact on the final product. CQAs identification is based on the severity of harm to the patients. Identifying high-risk CQAs during the pharmaceutical development is very critical. Let's understand by a couple of examples. When product contains two APIs and one API is in very small quantity, consider API ratio in drug product is 5% of API 1, 80% of API 2, and 15% excipients. Content uniformity becomes the high-risk CQA for the API 1, which is 5% of drug product. And blend uniformity is intermediate CQAs due to small amount of API 1 present in the drug product. Selection and control of CPPs during the blending operation are critical to achieve this CQAs. Another example is high soluble API is coated to control the drug release like extended release product. The dissolution percentage at different time point is the potential high risk CQAs for this kind of dosage forms. This is the example of the CQA. CQA is like characteristic of drug product should be within an appropriate limit range to ensure the desired product qualities. In this example, content uniformity is marked as a CQA as content uniformity is directly linked to the product safety, efficacy, and quality. So any variability in the content uniformity or out of limit may directly impact on the product quality. The differentiation shows that order is not the CQA. It could be the unpleasant and in terms of the acceptability by the patient, but it's not linked with product safety, quality, and efficacy. So that's the basic difference between CQA and not CQA. Here's the example of CPP. CPP is a process parameter whose variability has an impact on a CQA and should be monitored or controlled to ensure the process produces the desired quality product. Here's the example of size reduction CPPs. We have already discussed about the milling operation earlier. Any variability or any out of range variability of CPP could directly impact on the product quality and CQA may go out of limit. So this is the very critical to maintain the CPP within the range during the operation or pharmaceutical development. Example of CMAs. CMAs is a physical, chemical, biological or microbiological properties or characteristic of an input material that should be within an appropriate limit, range or distribution to ensure the desired quality of output material. As we have all discussed earlier, the criticality of selection of the right material with the right attributes, how it's linked with the critical process parameter and the critical quality attributes. Here are some example like particle size distribution, shape, particle or bulk or tap density, adhesive property, hardness, and so on. It is a science-based approach used in quality risk management outlined as per the ICH guideline Q9. Risk assessment is used to establish link between material attributes and process parameters for the final product CQA. It is a science-based approach to identify which material attributes and process parameters potentially have an effect on the product's CQA. To perform early in the pharmaceutical development process phase and required to repeat in various development phases depending on the product process knowledge increased during the development and the commercial phase. To identify and the rank parameter such as process, equipment, input materials with potential to have an impact on the product quality. This is based on the prior knowledge and initial experimental data. To determine the significance of individual variables and the potential interaction during the development phase. These variables and interaction can be further studies like throughout a combination of design of experiment, mathematical models, studies that lead to mechanistic understanding to achieve a higher level of process understanding. One of the example of risk assessment tool called Ishikawa diagram or fishbone diagram. This risk assessment tool required a cross-functional team of experts working together to develop a Ishikawa diagram. It helps to identify potential variables which can have an impact on the desired quality attributes. Risk rank the variable based on probability, severity, and detectability by using the risk assessment tool such as failure mode effect analysis or similar. It's also based on the prior knowledge and initial experimental data. It helps to develop control strategy based on identified higher risk rank variables. Example of risk assessment of tablet manufacturing as per ICH guideline Q8 appendix 2. This is the tablet manufacturing example. The different factors are like analytical, drying, plant factors, compressing, granulation, and raw material. There are many variables and the CPPs controlling these operations. For an example, drying, there are CPPs like temperature, relative humidities, airflow, shock cycle. These variables or CPP can have directly impact on the product quality if it found outside of the limit. Design space. Design space represents the relationship between the process input such as material attributes, process parameter, and the critical quality attributes. It is expected that operation within the design space will result in a product meeting the defined quality. Let's talk about selection of variables. The risk assessment and the process development experiments are helpful to identify the variables and their range. To include a detailed description, rationale for inclusion or exclusion of the process parameter and material attributes application in the design space and their effect on the product quality. Highlight process parameter and material attributes that were not varied through the development. This should include in the submission. Describing a design space in a submission. Describe through ranges of material attributes and process parameters, more complex mathematical relationship, time-dependent function such as temperature and pressure cycle of lyophilization cycle, or as a combination of variables such as components of multivariate model. Scaling factor to include if the design space is intended to span multiple operational scales. Analysis of historical data gathered throughout development phase and later study can contribute to the establishment of a design space. This information should include in the submission and supported by the relevant experimental data. Operation design space. To establish independent design space for one or more unit operations or a single design space for multiple operations. A separate design space for each unit operation is simpler to develop, whereas a design space that spans the entire process can provide more operational flexibility. Information on variables such as concentration, time, temperature are affecting the stability during the development and its control strategy should represent in the submission package. Relationship of design space to scale and equipment. A design space can be developed to cover a variety of scales. Consider the required operational flexibility. It is required to justify the relevance of a design space developed at a small or pilot scale to the proposed production scale manufacturing process. A detailed discussion on the potential risk in the scale-up operation is required to be included. Describe relevant scale-independent parameter where design space is applicable to multiple operational scales. Dimensionless number and/or model for scaling to include as a part of the design space description. Space versus proven acceptable range. Proven acceptable range based on univariate experimentation can provide useful knowledge about the process. The proven acceptable range is a characterized range of a process parameter for which operation within this range while keeping other parameters constant. This will result in producing a material meeting the relevant quality criteria. Whereas design space is the multi-dimensional combination and interaction of input variables and process parameters that have been demonstrated to provide assurance of quality. Working within the design space is not considered as a change. Design space and age of failure. It is to determine the age of failure for process parameter or a material attributes beyond which the relevant quality attributes cannot be met. However, it is not essential part to establish the design space. Strategy. Control strategy is very important in pharmaceutical development and manufacturing to ensure the consistently manufacture of a product that meet the required quality. To justify how in-process control and the control of input material like drug substance and excipients, intermediates in in-process material, container closure system, and drug product contribute to the final product quality. Control should be based on the knowledge of product, formulation, process understanding, critical process parameter, and material attributes. Identify source of variability by process, product understanding, assess impact on the product quality to implement the control strategy. Appropriately identified source of variability that provides an opportunity to shift control upstream and minimize the need for end product testing. Product and process understanding in combination with quality risk management supports the control of the process to adapt the variability to deliver consistent product quality. Control strategy enhances understanding of product performance, can justify the use of alternative approach to determine that the material is meeting its quality attributes. This alternative approach can be used to support real-time release testing and provide an increased level of quality assurance compared to the traditional end product testing. For an example, disintegration tests instead of dissolution test for fast disintegrating solid forms with a highly soluble drug substance, or dose uniformity performed in process by using weight variation coupled with the near-infrared assay. Real-time release testing can replace end product testing, but does not replace the review and quality control step called for under GMP to release the batch. Strategy can include, but it's not limited to the following. Control of input material attributes. For an example, drug substance, excipient, primary packaging materials. This based on the understanding and the prior knowledge of their impact on the process and the product quality. Product specification. Control for unit operation impacting on downstream processing or product quality. For an example, impact of drying on degradation, particle size distribution of granulate on dissolution. in-process or real-time release testing in lieu of end product testing. For an example, measurement and control of CQAs during processing. A monitoring program for verifying multivariate prediction model. For an example, full product testing at regular intervals. A control strategy can include different elements, and it could rely on end product testing or real-time release testing. Any of these elements can be, or alternative approach can be used by the applicant and provide the detailed rationale in the submission. Product lifecycle management and continual improvement. It provides opportunities to evaluate innovative approach to improve product quality. The detail on the product lifecycle management is explained in ICH Q10. The process performance can be monitored to ensure that it is working as anticipated to deliver product quality attributes as predicted by the design space. It includes trend analysis of manufacturing process as additional experience is gained during the routine manufacturing. Mathematical models, periodic maintenance could be useful to ensure the model's performance. The expansion, reduction, or redefinition of design space could be desired upon gaining additional process knowledge. Change of design space is subject to regional requirements.